Regenerative medicine, so we've been looking at multiple sclerosis. What do you want to do there? Well, they're really good immunosuppressive agents, okay? Galenia from Novartis, BG12 with Biogen IDEC. The new direction is regenerative medicine, repair, okay? So can you remyelinate? Again, there aren't very good targets for remyelination, so we set up a phenotypic screen where we simply screen for molecules that would induce oligodendrocyte precursor cells, it's a stem cell in the brain, to oligodendrocytes, which myelinate. So you're using, it's a stem cell-based therapy. All you want to do is make new myelin that's made by oligodendrocytes. You don't have enough oligodendrocytes in MS. That means you need to differentiate OPCs to oligodendrocytes. A very simple idea. So we got rat primary OPCs carried out a screen, found really kind of interesting novels. Those novels are going into preclinical, those are going into EAE models right now. They have, one of them has excellent brain exposure and it looks like it has good safety. We're testing its efficacy, but we also found known classes of drugs, antimuscarinics, that were active. And so these are really interesting because then we demonstrated that it was remyelination and not immunosuppression. Interestingly, we took those in combination with the S1Ps, okay, FTY720, which has cardiotox. And we showed when you do that drug combination of an immunosuppressive agent with a remyelinating agent, you can reduce in the EAE model the dose of FTY720 by a factor of 10. So you make it a safe drug, okay, which is important because the patent runs out in 2019. So you all of a sudden make it a 10 times safer drug, okay, but you remyelinate on top of it. So you potentially make it the best MS therapy in the world. So we're in active discussions right now with people in the MS space to actually combine immunosuppressive agents with remyelinating agents to both make safer and more efficacious drugs. And as I said, we also have novel compounds as well. So we have molecules that work out by completely different mechanisms that I think will also provide an interesting approach towards remyelination and repair. We're also taking a similar approach towards osteoarthritis. This is probably, one of the largest disease opportunities in all of the pharmaceutical industry, but it's also been one of the most difficult. How do you deal with osteoarthritis? Well, we took a repair approach. And so what you really want to do is form more chondrocytes in your knee joint to form more cartilage if you want to do repair. And so where do the chondrocytes come from? They come from MSCs, so simply get MSCs and screen for molecules that turn MSCs into chondrocytes. And what's the screen? The screen is actually you look for chondrogenesis and cartilage in the well. So you're looking, you have no idea what you're doing, but you know exactly what you're trying to find, okay? It's a totally validated disease phenotype. So we found molecules that do this, and they operate by interfering with a protein-protein interaction in the cytosol and sending a transcription factor to the nucleus that's a chondrogenic transcription factor. So even if I told you that was a validated target, you wouldn't think you could get a drug to do it. So we made, again, a lot of analogs of this molecule and assayed their activity. We've actually now got canine data, and the canine data is really quite impressive, okay? These are it's a surgical OA model in canines, okay? And you start treating after the OA is established, and you see you actually get significant decrease in cartilage degeneration score, okay? So it actually looks like a repair process is going on. And so we're really quite excited about this. This data is really pretty impressive. And again, the molecules, once they're out of the intra-articular space, this is an IA injection. Once you differentiate the chondrocytes, as soon as the molecule goes into systemic circulation, it's cleared, okay? So we can't really measure systemic concentrations of this drug. So the safety preliminary safety assessment looks really quite good.